of the vocabulary for Unit 6 cell division. The first three that you've got on your review sheet are forms of DNA. So we've got chromatin, which is thin and noodly. It's present during interphase. Chromatids are half of a chromosome, and then chromosomes themselves are the most condensed form of DNA. And this is how we organize the DNA after prophase to start getting it to opposite ends of the cell for division. The other part of a chromosome that you would have to know is the centromere. And the centromere is this button right here in the middle of the two identical halves of a chromosome. They are pulled toward the centrioles. Centrioles move to the poles, and they tell the cell how to divide things up equally. So if you pull half of it to this side, half of it to this side, that way we ensure that everything gets divided equally. The spindle fibers are attached there at the centrioles. Sister chromatids are identical copies. So you got one chromatid here from mom and one blue chromatid from dad. During interphase, each of those doubles. So now you have two identical chromatids from mom. We call those sister chromatids. You also have two identical chromatids from dad, sister chromatids. If you look at all four of those together, the two from mom and the two from dad, that's called a tetrad. Four means tetra. Tetra means four, sorry. And so when you talk about two sister chromatids from mom, two from dad, that equals four. We call that a tetrad. And that is most relevant during interphase, prophase, metaphase of meiosis 1. We call it a tetrad. When we talk about diploid and haploid, it's just referring to the number of chromosomes. Your diploid number is the number of chromosomes that we see in your body cells, where you have 2 of every chromosome for a total of 46. You'll remember that from your karyotyping maps that you did online. Haploid number is how many chromosomes you would have in your sex cells. You should have just 1 of each chromosome for a total of 23. Cycle, beginning with cytokinesis of mitosis. Right after the cell splits, it goes into G1, which is its first growth phase. Cell's just doing life, being a little muscle cell, a little nerve cell. And then it goes into the S phase, which is where it copies its DNA and doubles its DNA to prepare for division. G2 is the second growth phase, where the cell is going to increase in size, double the number of organelles and things inside of it, and then it's going to be ready to divide during the M phase. M stands for mitosis. That's when it goes through prophase, metaphase, anaphase, telophase, and cytokinesis to actually split that cell. If you add up the first 3 phases, G1, S, and G2, all 3 of those added together make up interphase. So when we say that interphase is where cells spend most of their life, that's because they're doing all 3 of these during interphase. There is a checkpoint at the end of each of these parts of the cell cycle. The G1 checkpoint simply says, did that cell grow and live as a healthy cell? If so, it's allowed to continue. If not, that cell is supposed to die because it might be cancerous. At the end of the S checkpoint, after we've copied DNA, we have another security check that just says, did that DNA copy correctly? If so, go ahead and continue. If that DNA did not copy correctly, then it's supposed to die. G2 checkpoint, remember we're checking to make sure that it has grown enough to split up equally between the 2 new cells that it's about to create. And then the M phase checkpoint is where we're looking at those chromosomes at metaphase and making sure that they're all lined up. We did our buddy check. Nothing's missing. We have nothing extra. And if that's allowed, then the cell continues to divide. And if it is not passing that checkpoint, if there's something wrong with those chromosomes and how they divided, that cell is supposed to die. Cancer cells avoid all of this because they are not stopped by the checkpoints. They just keep dividing over and over and over again, which makes really large tumors and causes cancer cells to spread even though they have a lot of problems. Of making more body cells. So the phases here involve interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. You guys designed a lot of creative mnemonic devices. The one I've always used is, I puked on my algebra teacher's car. You could also say, I probably made a teacher cry, or inside people's minds are the conclusions. I think that was one of them. But anyway, in interphase, you see that the DNA is all kind of loosely scrambled up, kind of looks like spaghetti noodles. That's chromatin, and the DNA is going to double. Prophase, all of that DNA is going to condense into those little X-shaped chromosomes. The nuclear membrane goes away. In metaphase, we're going to line up all of those chromosomes in the middle. Spindle fibers are coming out from the centrioles to line them up. Anaphase, the spindle fibers are pulling those separated chromatids toward the centrioles to the opposite sides of the cell. During telophase, those chromatids are going to get wrapped up into new nuclear membranes. So you can see we're forming two new nuclei, and the chromosomes themselves are starting to relax. And then cytokinesis is different in plants and animals. In animals, we get this pinched in section here, and we call that a cleavage furrow, that point where it pinches in. But in plants, they build a cell plate, which is a new cell wall that comes down the middle of the cell, and that's going to split those cells in half. Meiosis looks similar with a couple of key differences. In meiosis, we're trying to make sex cells. So we have two goals here. We have meiosis 1, and the goal is to scramble up that DNA, scramble up the genes that are passed along from moms and dads through the process of crossing over. During meiosis 2, our second stage of division, we have to cut the number of chromosomes in half by dividing each cell a second time. So we're creating 4 cells at the end of meiosis instead of just the 2 cells that we had at the end of mitosis. So what we've got here, interphase, the chromosomes that were present, 46, they double. Now we have 92. Prophase and metaphase, we have crossing over, so it's the exchange of genetic information. Anaphase, telophase, and then at the end of cytokinesis, we now have 2 cells that do not look the same. Because of that crossing over, they're no longer identical, and we're back down to our starting number of 46 chromosomes. Now we immediately go into prophase 2. And during prophase, metaphase, and anaphase 2, we are splitting that DNA in half again. So what we're left with at the end of cytokinesis 2 is 4 absolutely unique cells, and each of those has half of the amount of DNA that we started with. That is important because, as we'll find out later, if these are going to be sperm and egg cells, each has to contribute half of the genetic information to the baby. If we have too much genetic information, we have extra chromosomes, which is a problem. Difference between mitosis and meiosis. Mitosis is all about body cells. We go from 1 cell to 2, and both of those new cells are identical with the same number of chromosomes as what you started with. In meiosis, we are trying to make sex cells. So 1 cell is going to go through 2 separate splits so that we end up with 4 cells total, and all 4 of those new cells are genetically unique, and they do not have the same number of chromosomes. They've got half the number of chromosomes as the cell you started with. Necessary because we want exactly half the genetic information coming from mom and her eggs and half the genetic information coming from dad and the sperm. So because of that, we need just 23 chromosomes for humans coming into each of those sex cells. Crossing over kind of looks like the picture I've got here. So a blue chromosome represents the double you got from mom, the white represents the double you got from dad. When they line up at metaphase, they just exchange little pieces of DNA across those chromosomes, and that way you are getting a mixture of what your parents gave to you to pass along to your own children in your sex cells. So meiosis and crossing over occurs during prophase 1 and metaphase 1. Meiosis looks really, really complicated, so there are some advantages, but also a lot of disadvantages for an individual who can reproduce that way. So if a cell is only reproducing through mitosis, this would be something like a bacterial cell or a yeast cell, or even some very simple worms. They can do this really fast, and because of that, they can make a lot more babies, because all they have to do is split themselves. They do not need to go find a mate, but the problem is that they have a lower survival because they are all clones. So if something would be harmful to one, it is harmful to all of them because they have no genetic differences. Meiosis is what you and I and all the other animals, pretty much all other animals, do for reproduction. And because we have all of that mixed up DNA that occurs through crossing over, we have more diversity and better survival. We are not all clones, so just because you are allergic to peanuts doesn't mean I'm allergic to peanuts. It is not great because we have to spend a lot of energy to find a mate. It is a slower and more complex process, and there's more chances to have an error when you have a complicated process. During our discussion in class, but a stem cell is like a blank slate. It can be turned into anything because they haven't yet been assigned a specific job within your body. They haven't been assigned to be skin or muscles or nerves. So we can program them to do any of those things and to restore those things. So we can get paralyzed people unparalyzed basically by restoring their nerves. We can treat or cure diabetes, cancers, Alzheimer's, Parkinson's disease, a lot of other things like that. And we can do that without using drugs, medicine, and advanced surgeries because it's coming from within. It's coming from the cells themselves. However, it is expensive. People have ethical problems with where we are getting these stem cells. And having stem cell treatments might actually cause certain cancers. Haploid and diploid numbers of chromosomes, remember you're just talking about if we are talking about 2 copies of every chromosome for diploid or 1 copy of every chromosome for haploid. Haploid numbers are found in sex cells, diploid numbers are found in body cells. If you were to calculate that for humans, our diploid body cells have 46. After interphase, they duplicate. So prophase 1, they've got 92. They split. So metaphase 2, they only have 46 in each of those 2 cells. And then they split again. So at the very end of meiosis 2, we're down to just 23 chromosomes. We follow that same pattern for all individuals. Karyotype, which is just a map of the chromosomes that we would find in a person's cell. What you're looking for first is what sex is this person? And to do that, you look at the X and Y chromosomes. If they have an X chromosome, they're female. If they have a Y chromosome, they are a male. So no matter what else they have, a Y makes a guy. A disorder when you have an extra chromosome for a total of 3 of that chromosome type, we call that a trisomy. That prefix tri means 3. So there's an example right here on this karyotype of a female, 2 X chromosomes, with a trisomy of chromosome number 13. So this chromosome has an extra copy. There's lots of examples of this, including trisomy 21, commonly known as Down syndrome, or XXY, where you have 3 sex chromosomes, and that's called Klinefelter syndrome. However, there are also cases where you are missing a chromosome. So instead of having a pair for each of these sections, you have one that's missing. We call that a monosomy. And an example of that, there are very few that people survive, but one example of that is where you have one X chromosome, but nothing else. You don't have a second X or a Y chromosome. We call that Turner syndrome, and that's one of our very few examples. This happens through a process called non-disjunction, where those chromosomes are not equally divided during meiosis 1 or meiosis 2. If that error occurs during meiosis 1, the first split, it's going to mess up everything after that point. So most of the cells are going to have the wrong chromosome number. If the meiosis 2 non-disjunction occurs, half of the chromosomes should be right and half the cells should have the wrong chromosome number. That's the worksheet that we did in class just yesterday. So if you're having trouble with that at all, go ahead and look back at that worksheet that we talked about non-disjunction, where we talked about meiosis 1 and meiosis 2 problems. Good luck on tomorrow's test, and I will see you tomorrow.